We have started our experiment with the proteomics analysis. So first we have validated whether our samples are biologically reproducible or not using this PCA plot, and we can see that biological no, biologically biological replicates are uh, biological replicates are close to each other. So it represents that our sample preparation is good and is biologically reproducible. And for preparing this PCA plot, we have used top 50 variable protein, and with the normalization, considering the samples are prepared on different days and belongs to different batches. And simultaneously, we also get the information that they each knockouts are behave in a different manner and compared to the wild type and also compared to each other. So we try to analyze some of the highly dysregulated proteins in individual knockout so that we will get some insight about the changes that are going on within the cells. So in this figure, it is the volcano plot is represented about this calnexin knockout compared with the wild type. So when we check the highly upregulated proteins within the calnexin knockout, we can see that we can see that some of the proteins that are highly upregulated has provided us some insight that is going on within the knockouts. For example, this ATP13A1 proteins that is the protein that is produced using this ATP13A1 gene plays a role in preventing the cross-contamination of mitochondrial proteins into the endoplasmic reticulum or endoplasmic position location. So upregulation of this could indicate that in case of calnexin knockout, there could be a contamination of the proteins from mitochondria to endoplasmic reticulum. That's why the upregulation of these proteins is trying to reduce that compensation. And then second protein that is highly upregulated is related to this NDB52 gene, and this protein is actually this protein from the literature. Uh, in the literature, it has been reported that this protein is related to autophagy receptor. So it normally acts during the viral or bacterial infection. So whenever the cell experience any viral or bacterial infection, these autophagy receptors get activated. So this could also be one of the reason why we do not see high VLP production um, during the experiment the Chi has conducted or Chi has reported in the Science Advance. And another interesting thing that we have observed is about these proteins, that is GTSE1, and the literature. It has been reported that GTSE1 is related towards preventing the apoptosis within the cells. So this could indicate that this high production of GTSE is somewhere related to is somewhere related to um, survival of the cells or preventing the apoptosis within the cells. And when it comes to the downregulation proteins, we see the highly downregulated protein, that is WTAP, and this is also related to this regulator of antiviral response. And uh, the one of the reason of this WATP downregulation is that this IFN1 is upregulated, and this IFN1 is uh, upregulation of this IFN1 is related to this antiviral response. So in calnexin knockout, the high IFN1 upregulation will prevent the viral production, will prevent or will cause this antiviral response that will prevent the VLP production in this calnexin knockout. And these two uh, observations are in line with the result are in line with the results that is published in the Science Advance. That is, to we do not see any signals for VLP in the calnexin knockout. And other two proteins are downregulated are this related to this gene, that is EPHB2 and AR2 gene. So these proteins are the glycoproteins. One of them is transmembrane glycoprotein. This downregulation of this indicate that this calnexin or the downregulation of this glycoprotein indicates the role of calnexin in this glycosylation pathway. So absence of calnexin will leads to less production of this glycoprotein, as uh, some of these glycoproteins might move towards the ERAD pathway, might move in the ERAD pathway because they are not able to acquire their native structure. And simultaneously we did this study on other knockouts also. So in brief, in this calnexin and MOX knockout, what we have find out that the proteins that are related to UPR response that usually triggered when there is a ER stress are upregulated. And also in case of this calreticulum knockout, the other chaperones are also other chaperone, that is PPIB is upregulated. So this upregulation might be to compensate the role of this calreticulum, whereas this upregulation of this protein is not seen in other knockouts. And the the glycosylated proteins, both N and O-linked glycosylations are downregulated in uh, both the knockouts. And in case of dual knockout, we have seen some unexpected result in a way that there is a down regulation. It's not up regulation. There is a down regulation of. We have seen some results such as there is a down regulation of glycosylated proteins, uh, and these are heavily glycosylated proteins that has around 15-20 glycosylation site. The first one is this insulin, and then this GNA 13. And however, we do not see any UPR related proteins. So this is surprising, as we are expecting the high UPR response, because in this case of dual knockout there is an absence of both the calnexin and calreticulum. So there will be no calnexin calreticulum cycle. And one of the reasons could be that uh, since there is no calreticulum and calnexin cycle, the protein's residence time within the endoplasmic reticulum decreases, and they will directly move towards the ERAT pathway. And 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 the cellular optimization proteins, that is HMG11, is also down regulated are also down regulated. So this indicate that because of the down regulation of this protein, the cells might not be able to perform its function in a optimized cell might not be able to optimize its performance. So this could be one of the reason why cells are not behaving in a logical manner. So what are the main findings that we observed from the proteomics is that there is a UPR response that we can see in some of the knockouts, but we do not see in any other knockout. Do not see any UPR response signals in the dual knockout. In calnexin knockout also, we can see some upregulations. In calnexin knockout also we can see some upregulations. Whereas in dual knockout we do not see any upregulated UPR proteins, and some of the glycosylated related proteins are also dysregulated. And finally, we have seen the apoptosis suppression that is observed in the MOX and calnexin knockout. So first we try to verify our observations, that is about UPR response, and that we did it using UPR functional assay. So how this UPR response work is that it generates three major signals. First one is that ATF6, another one is PERK signal, and third one is IRE1 signal. So whenever the cells observe a ER stress, it creates these three signals that will generate UPR response. So in case of ATF6, what happens is that this ATF that is usually present on ER membrane will move to the Golgi, and the proteolytic cleavage occur, and one part of this ATF6 will move to the nucleus, and it will upregulate, it will upregulate the proteins, upregulate the translation of the proteins that are involved in reducing the UPR response. That is upregulated the ER chaperones, that is calnexin calreticulum, that can take care of the unfolded proteins that are increased within the cells or within the endoplasmic reticulum. And similar thing is done by the PERK signal also. So in this case, this phosphorylation of this EIF2 alpha occurs, and this will trigger this ATF4 uh, this will trigger this ATF4 translation, and then this ATF4 will move into this nucleus to trigger the proteins that are involved in autophagy or redox, or we can say that amino acid biosynthesis. So the main thing is that the autophagy is directly or indirectly related to this ERAT pathway. So it will take care of the proteins that needs to be degraded, unfolded proteins that needs to be degraded. The third one is this IRE1 alpha. So in this case, what happens is that this XBP1 mRNA, it is in normal condition, there is an intron that is present that prevents the translation of this XBP1. However, in case of this dimerization of this IRE1, this intron, this it will help in removing the intron of this XBP1, that will uh, that will leads to the translation of this XBP1, and the XBP1 will also upregulate the proteins that are involved in ERAD or ER chaperones. And in literature, we are able to find two reporters uh, that from which that can be used to measure whether the cells has the ER stress or not. So we have used this ATF4 reporter and XBP1 reporter. So these are the two report. These are the PERK reporter. How it works is that in case of when there is no ER stress, the ORF1 and ORF2 region will be translated, and this ORF3 region will not be translated. However, whenever there is a ER stress, the ORF3 region will be translated because of the epigenetic changes because of the ER stress. So whenever this ORF3 region is translated, we are able to see this red signal because of the M scarlet. And in case of this IRE1, whenever there is no ER stress, translation will stop just before this green signal, and whenever the cells observe the ER stress, this intron part is removed, that will leads to shift of base pair, and now the translation will end after this NLS sequence. So during this case, during this ER stress case, we will get the green signal whenever there is IRE1 signal. IRE1 signal is triggered. So first we did the UPR assay on the wild type to check whether we are able to get a bright signal or not. And for this, in order to generate the ER stress, artificially generate the ER stress, we have used thepsigargin. It will disrupt the circa pathway of ER. That leads to the ER stress generation, and that will trigger the UPR response. So this is how we did the experiment. It's a 2-day experiment. For 1 day, we have added this lentivirus that contains this reporter, and on the second day, we have, just before this uh, fluorescence measurement, we have added the thepsigargin to generate the ER stress. And in these signals, in this image, these fluorescence images, we can see that signals for the perk reporter are bright, whereas compared to the signals for this IRE1 reporter. And and these images are for this 800 nanomolar thepsigargin concentration. When we quantify these images on the time base, we can see that the perk reporter signals, this is for the perk reporter. Perk reporter signals are bright, and we can see the difference between the control and this negative control and the positive control uh, easily. Whereas in case of this IRE1 reporter, we can see the difference in the negative and the positive control. However, the difference is very low. So because of that, we decided to move with this perk reporter to do the functional studies on the knockout. So in these functional studies, what we did, we first prepared stable cell lines, and also selected our cells using this pyromycin, so that we will have the cells that contains that we will have the cells that contains our fluorescence reporter, so that we can get the more bright signals. So we can see that in this case, in the case of this dual knockout, uh, this calreticulum has very bright signals, and calnexin in these images, it looks like this calreticulum calnexin signals are quite similar, whereas for MOX the signals are low, where and for dual knockout signals are least. When we quantify these images, we can see that for calreticulum, the ER stress is very high. However, for this calnexin and MOX, the ER stress are comparable. So this two, for calnexin and MOX, the ER stress are comparable. And for this wild type and dual knockout, and for this dual knockout, the ER stress are almost negligible. This is in line to the InQSight images. And when we and this is just the quantification of these fluorescence images, and we also did separate parallel studies, parallel measurement using the flow cytometer, and we have found out the similar results. That is calreticulum, the ER stress is very high, and for this calnexin and MOX knockout, the ER stress are comparable, and for the dual knockout, ER stress is very low. And so these signals are based on the fluorescence reporter. However, we also did a independent study using this CHOP protein. The CHOP protein is usually produced whenever the cells observed a ER stress. So when we did the CHOP protein when we analyze this CHOP protein using Western blot, we can see the results that are in line with the flow cytometry and InQSight results. That is, in calreticulum knockout, protein CHOP protein production is high, whereas in calnexin MOX knockout data is low, and for the dual knockout we do not see any, we see very low signals for the dual knockout. And so this is for the UPR response. Next observation that we get is the some glycosylation related proteins are dysregulated. So this is in line with our initial hypothesis that glycosylation profile will get dysregulated. So for this we decided to do two experiments. First one is the cell surface glycan study, and next one is the whole cell glycomics. So in cell surface glycan study, we use 23 different lectins that are available in our lab to check whether there is a cell surface change in the cell surface glycan structure or not. So we have used flow cytometer to measure these signals, and these represent the normalized geometric mean data. In this, we have considered the wild type signals as 100. So what we have observed in this case is that only in 4 cases there is a significant change in the signals, and these all 4 changes are in calreticulum knockout, that is in PNA, RCA, GSL2 and AAL. And another interesting thing is that all these 4 signals belongs to a different kind of glycans. That is, PNA belongs to O glycan type, RCA belongs to galactose, and this GSL2 belongs to glycnac, and AAL belongs to fucose type. So these other lactanes, the difference between these 3 lactanes, for example, is that within fucose type they will bind to the different motifs. Uh, that's why we do not see signals for other 2 lactanes, whereas we see signals for specific lactanes. So this also provide a insight that there is some kind of glycosylation profile changes occurs in the calreticulum knockout, and this could be one reason, could be that there is a quality control compromisation or quality control degradation occurs within the endoplasmic reticulum uh, in the calreticulum knockout, in the calreticulum knockout. Next we did the whole cell glycomics. So in case of whole cell glycomics, we have analyzed the intracellular glycans and also the cell surface glycans. So this is the experimental plan that we followed. We take out the cells and remove the glycans, release the glycans using PNJ7 and run it on PGS column. So when we check when we do this upset plot analysis, we are able to find that in case of dual knockout there are 26 new glycans or 26 unique glycans that are only present in the dual knockout but not in any of the other knockout. So this could indicate the point that in case of dual knockout there is a high quality control degradation, and most of these these glycans could be within the uh, within within the cytosols, because we do not see any within the cytosols or within the cells. We can say that because we do not see any significant changes in case of dual knockout on the cell surface glycan motifs. However, for this MOX knockout and this MOX knockout and calnexin knockout, the unique glycans are around 11 and 10. So these are comparable, and this is also in line with our ER stress result, where we see the glycan structure, where we see the glycan structure, where we see the ER stress are also comparable between this MOX and calnexin knockout. And in case of this calreticulum knockout, there are only 3 unique glycan structure. So this provide a better picture about the uh, quality control uh, quality control within the different knockout cells. And since there are only 3 unique glycan structure in the calreticulum knockout, this provide us This provides us the information that in case of catalytic knockout, there could be less quality control compromise compared to the other knockout. And the changes that we have observed in case of this dual knockout could be because of the change in the hybrid and complex structure. As in case of wild type, we can see that hybrid and complex structure are almost comparable, whereas in case of this dual knockout, the complex structure is increased and this hybrid structure has been reduced. And similarly, the other changes are also, most of the changes are related to this hybrid and complex structure in case of other knockout also. As we can see that the hybrid and complex structure compositions are different compared to the wild type. Whereas in case of calreticulum knockout, there is a increase in the amount of core fucose also. So core fucose structures are more in case of calreticulum knockout. So the fucose structure are the ones that fucose structures are more probable of passing through this calreticulum knockout compared to the other structures. So this is what we have just now discussed. And last thing that we have done is this apoptosis assay. So although we have already planned to do this apoptosis assay before we got this proteomics result, however the proteomics results also suggested that some observations that we are expected to see in this apoptosis assay, as we have seen that apoptosis suppression proteins in this MOX and calnexin knockout. So we decided to do two studies as per our initial plan. First one is the apoptosis assay and next one is the cell cycle assay. So when we did the apoptosis assay, we do not see any significant changes in the dead cells that are present in the different knockout. So, and when we quantify these results for two technical replicates, we can see that the apoptotic cells are in line with the wild type. So for this case of for the case of this calnexin and MOX knockout, we have seen upregulation of apoptotic suppression gene. So that would explain that why we do not see any apoptosis in this calnexin and MOX knockout. And in case of dual knockout, we do not see any UPR response. So that could explain that why we do not see any apoptosis in this case of dual knockout. However, when we check for calreticulum knockout, in this case, why we do not see any apoptosis, one of the reason is that the ER stress behaves in a way that it will either try to restore the cells to its normal condition, or if the ER stress has crossed beyond a threshold level, then it will trigger this apoptotic assay. So in this case, the ER stress could be beyond the threshold limit. That's why the apoptotic signals is not triggered. However, why we do not see any upregulation of GTSE is not clear. Maybe since the ER stress is within the threshold limit, that's why we do not see any upregulation of this GTSE gene. So, yeah. Next, we did the last experiment we did is the cell cycle assay. So, when we did the cell cycle assay, uh, here also we do not find any changes within the cell cycle. Almost all the cell cycles, almost all the knockouts behave similar to the wild type. And when we did the statistical analysis also of the two technical replicates, we do not see any significant changes. So So what is the final conclusion that we get from this overall study is that for this, let us start to answer our first motivation that we have before the beginning of before let us try to answer our first questions that we have before the start of this project. That is why we see no VLP production in the calnexin knockout and high VLP production in the calreticulum knockout. Because in case of calnexin knockout, there is a upregulation of two proteins related to this WTAP gene and this NDP52 gene, and both of these the proteins that are formed from both of these genes uh, his focus is on try to reduce this virus production within the knockouts. So combined effect of this could be the reason why we do not see any upregulation of any signals about this VLP in the calnexin knockout, whereas no whereas these genes are neither shown in this case of calreticulum knockout. That's why we see high production of this gene VLPs in the case of calreticulum knockout. And another thing, but for which we don't have any evidence, that since the production of VLP is high in case of calreticulum knockout, it might not be uh, interacting with the calreticulum directly, or this VLP does not need the help of calreticulum in order to form its native structure. That's why uh, the VLP production in case of calreticulum knockout has increased even has increased significantly compared to the wild type. And for the case of this calreticulum knockout, this high ER stress in calreticulum knockout uh, could indicate that this calreticulum knockout has a broad specificity, or calreticulum knockout take care of interacts with a large amount of proteins since it is a ER lumen protein, whereas calnexin is is a ER membrane protein. So it can probability of calreticulum proteins to be available in the endoplasmic reticulum is more compared to the calnexin, and this results also indicates the same thing, that in case of this calreticulum knockout, knockout, cells observe a higher ER stress, as the other chaperones are not able to compensate the role of this absence of calreticulum. And for the case of this MOX knockout, we have seen the down regulation of glycoprotein genes and upregulation of this ERAD pathway genes in case of MOX knockout. So this, since this protein is absent, so because of this, what happens is that now this glycan structure is not detected by the calnexin and calreticulum cycle. So since it's not detected by the calnexin and calreticulum cycle, these proteins will not be able to form their native structure, and they will be moved to this ERAD pathway. And one more thing that might happen is that since they will move to this ERAD pathway directly, as the residence times by these newly folded proteins that are not recognized by calnexin and calreticulum cycle will be reduced, as they will not spend much time in the endoplasmic reticulum compared to the wild type, since they do not interact, or they do not interact with the calnexin and calreticulum cycle. So this could be the reason why we see this upregulation of this ERAD pathway or ubiquitation proteins in case of MOX knockout. In case of MOX knockout. And for this case of dual knockout also, the hypothesis or results shows the similar trend. That is, we have seen the more unique glycans in the intracellular parts of the cells. So one of the reasons could be that since this calnexin and calreticulum are not available, so even this monoglucosylated structure is present because of the absence of this calnexin and calreticulum cycle. This there is an increase in the amount of this unfolded structure, and some of them could be diverted to this proteasomal degradation. However, the with time the unfolded structure within the endoplasmic reticulum is increased uh, to an extent that they will uh, bypass this quality control. They will bypass this quality control of this endoplasmic reticulum and move into the intracellular parts of the cells. That's why we have observed more unique glycans, more unique glycans in case of dual knockout. So, and also in case of dual knockout, since there is no calnexin and calreticulum cycle, these proteins might spend less time, or they will have less residence time in the endoplasmic reticulum. So before so since they have less residence time in the endoplasmic reticulum, so because of that they will move quickly uh, into the intracellular parts of the cells or to the proteasomal degradation, and this could be the reason we do not see any UPR response. And this observation is in line with with some previous literatures that also reported the quality loss in the case of dual knockout. So before we move on to the question and answers, I would like to acknowledge the support and help provided by the students and the professors. So the Chiang and Thomas has provided me the single knockout cell lines that they are using it for their own experiment, and Thomas also helped me in providing this Syn4H analysis or extracellular glycomics analysis. This is under process. That's why I have not presented these results in this thesis. And Jiang also helped me in developing the double knockout or dual knockout, and the lab in the downtown, or more specifically this Sichen Sen or Kevin and Ming Zhang help from the Q lab helps for this proteomics LCMS sample run, and then Professor Rudy Anto and Ejas helped me to do bioinformatics from these proteomics results. So that's why we are able to make the hypothesis that there is a ER stress, differential ER stress within the cells. And then this Sabrinath, and then the Sabrinath Subramaniam is the postdoc in Nebraska, University of Nebraska, and the professor Rebecca Gundry from the University of Nebraska Medical Center helps in the glycomic sample preparation and LCMS run. And then Gabby uh, from Professor Nilamingham's labs helped in using this glycomics toolbox that helps me to analyze the results from results that we have received from the University of Nebraska Medical Center. and and then Jesse helped in running the NGS data so that we are able to confirm that our knockouts are bona fide. And finally, the professor Shriram Nilawingams. So without his guidance and support, I will not be able to complete this project. Now, yeah, that's all for this project. Thank you for your time.